Welcome to Anatomy and Physiology 1. My name is Miranda Chandler. I'm your professor for this course. Your required textbook is the image you see on the first slide, the Visual Anatomy and Physiology. If you've not already done so, please order it online, reach out to the bookstore. There's online versions as well as physical versions you can obtain. You'll also be required to have an access code for MyPearson, which is an online platform where all of your homework assignment and your quiz assignments will be available and with all due dates required. Also, refer to Canvas, download your syllabus, your schedule of activities. This has all the important information, due dates, what we'll be covering each week in class. Please definitely download that and refer to it frequently. Please also reach out to me via email. I'm here to help you with any questions or concerns. Our first section we'll be covering is an introductory section. It covers chapters 1, 2, and 3. It's an introduction, basic chemistry, cell structure, and function. Okay, let's dive right into chapter 1. Chapter 1 is the introduction to this course. It just helps you have tips about being successful in this course. So how to be successful in this course? Read your syllabus, understand the grading policies, know how much a test is worth versus homework and quizzes. You will have graded homework and quizzes. You will have 5 unit exams and 1 final exam. The lowest homework grade, the lowest quiz grade, and the lowest unit test will be dropped. The final exam cannot and will not be dropped. Late assignments will not be accepted. All work is due on the specific date it is due. I allow you to work until 11:59 p.m. for that due date. So please mark your calendars, check your schedule of activities for those due dates, and be aware that they are due by 11:59 with no exceptions. Partial credit will not be accepted for any late work. It will receive a 0. Attendance, our class is online, so attendance will be kind of different. There will be online discussions. There will be obviously periodically, making sure your homework and your quizzes are done. That will count as your attendance. Attendance is not graded, however, it is definitely checked upon because I want to make sure that you are participating in the class, getting the most out of the class. The more you participate, the better you will be familiar with the material, the easier the material will come to you, and when it's time to take those tests won't be as difficult because you've put the effort and put the time in. Extra credit, sometimes I give extra credit, sometimes I don't. It just typically depends on how well I feel you guys are doing, and so it's a base by base situation. So it comes and goes. Don't count on it. Don't count on that extra credit because I may not give it. So, and it's usually only a couple points here and there. It's not enough to make a huge difference in your grade. So if you're coming into the final with an F, don't expect to get extra credit to bring you up to a B. It's just not going to happen. Know the policies and procedures and plan accordingly. So the exams are going to be via honor lock. So typically you're going to have roughly 3 days to complete that. I strongly recommend don't wait till the last minute because the exams are offered for 3 days. The makeup policy for exam, there's only 3 excused reasons to have a makeup exam. All other reasons are not excused. The reasons are jury duty, medical illness with a medical note, and military duty. So work doesn't count, you going on vacation doesn't count, the internet went down doesn't count. So those 3 are the only reasons for excused and makeup policy for the tests or homework or quizzes. So please plan accordingly. Check that syllabus, check that schedule of activity for those dates, and make sure that you're available and you're able to take those homework, quizzes, and tests on time. Print a copy of your schedule of activity and check it every day. Actively study 8 to 10 hours per week, roughly 2 hours per day, and you can multiply that by 2 for the summer session just because it's half a semester and it goes by really, really quickly. My recommendation is to kind of break that study session up. Don't sit down for 2 hours every day and study unless that works for you. For me, sitting down 10-15 minutes at a time, break it up into multiple sessions throughout the day, I tend to retain more information because I get fidgety, I get bored, and my mind wanders. So do what works best for you. Study in a group, study with flashcards, have family members quiz you, have friends quiz you, take online quizzes, watch videos, whatever works for you in studying. Continue to do those, and the information will be embedded in your brain. Attend every class and take notes during lecture. Slide decks do not represent all content you are responsible for. Don't forget, in college, you're expected to learn content independently and also to come in with foundational knowledge from previous courses. So, all the information that I present to you is typically on the exam. However, every word that I say is not going to be in that slide deck. So you watching the videos, because this is an online course, watching those videos, taking notes, referring back to your textbook, watching the other videos that are available in Canvas that I'm providing for you are going to help reiterate that information and solidify that knowledge that you're needing to know for those exams and for continuing on into AP2 and anything further in your medical career if you choose to continue in the medical career. Read your book. Use the artwork to understand the text. Use the guided notes to interact, engage with texts and lectures. So there are guided notes available for each chapter. They're basically a really big glorified study guide. I've had students do them in the past. I've had students not do them in the past. They do take a lot of time. It is a lot of effort. However, the students who typically do them, review them, use them to study for their exam, tend to do really, really well in the course. Create flashcards using the unit study guides, studying groups, but not social groups. So you want to study. You don't want to sit there and chat about what you did this past week. You want to actually study the material. Tips on how to succeed in your AP course: approach the information in different ways. So, like I said, watch some videos, take some online quizzes, use flashcards, use study groups, whatever works for you to help study better. Set up a study schedule and stick to it. Devote a block of time each day to your AP course. Practice memorization because AP is a lot of memorization. Avoid shortcuts. There is no shortcuts in AP. There's no little tricks that are going to help you remember everything. You have to put the time and the effort in. Attend all lectures, labs, and study sessions. Read your lecture and lab assignments before coming to class. Do not procrastinate. That is the one thing that, as I've been teaching, if people procrastinate and wait till the last minute, they typically are not doing well in the class because they're not putting the time and the effort into it. Seek assistance as soon as possible if you have any problems understanding the material. Reach out to me. That's what I'm here for. I'm here to help you. I'm here to help you succeed and to improve. So please reach out to me and I will help you in whatever way I possibly can. Okay, so introduction to anatomy and physiology, homeostasis, and anatomical terms. That's what we're going to be covering in this session. All right, so anatomy and physiology. Anatomy is the study of a structure. So here we have the picture of a heart. So in this example, anatomy is the actual structure of the heart. What is the heart made up of? What does the heart look like? What vessels are in the heart? How many chambers does the heart have? Those are the things that anatomy is. What can we physically touch and see? So we have a couple different types of anatomy. We have gross anatomy and we have microscopic anatomy. So gross anatomy is physically what we can see with the naked eye. When we dissect something in the lab and we're just physically looking at it, that is our gross anatomy. Our microscopic anatomy is what we can't see with the naked eye. We need the assistance of light or electron microscopes to see those fine details, to see those little tiny cells that make up that muscle tissue of the heart. So the difference between the two is basically large versus small. So with our different types of microscopes, we have the light microscope, which can see the basic details of the cell structure. It's the pretty much the microscope we use on a regular basis in our lab. We have an electron microscope, which can see individual molecules that are only a few nanometers across. And we have our dissecting microscope, which can see the tissue structure. So it's not a very high magnitude microscope. And you're going to get in more in depth in the different types of microscopes in lab. We're just kind of touching base on it in lecture. Anatomy and physiology is very heavy on memorization. Throughout the course, you're going to be given a lot of quote-unquote vocabulary terms. There's no easy way, like I've said before. You just have to memorize those terms and know what they mean. Physiology is the study of function. So anatomy was the study of structure, and physiology is the study of function. So the anatomy is the heart, the chambers of the heart, the atrium, the ventricles, where the aorta comes into the heart, the valves, the mitral valve, the aortic valve, how they open, they close. So the physiology is the study of the function. What happens when the heart squeezes? What happens when the heart relaxes? So that is what physiology is. Physiology, study of function and how living organisms perform their vital functions. Once again, this is very heavy on conceptual understanding. It's something we physically, yes, we can see the heart squeezing, but how do we know when the heart squeezes, what does it do to the rest of the body? So, for example, when the heart squeezes, that is how forceful the heart squeezes tells us our systolic blood pressure, which is the top number when we get our blood pressure checked. When the heart relaxes, that's our diastolic blood pressure. That's that bottom number on the bottom of our blood pressure. So understanding this conception of, okay, what happens when? Okay, so just to recap, just a quick little kind of put this in your brain because this is going to help you going forward with anatomy and physiology in general. Anatomy is to blank as physiology is to blank. So anatomy is to structure as physiology is to function. So kind of keep that in your brain because as we go through, you're going to ask yourself that question. Which is structure and which is function? All right, so reiterating that structure versus function. So structure determines the function. So, for example, we have the elbow joint here. This is the picture of the elbow joint. So we look at the elbow and we go, okay, well, the structure of the elbow, the structure of the forearm, the structure of the humerus, how do they interact together? What can their possible function be? So looking at that elbow joint, it functions like a hinge. A hinge is like the door. It opens and it closes, but only in a forward and a reverse direction. It won't wiggle side to side unless the hinges are broke. And it opens and closes. So that's the only function of that door. So functions like a hinge, moving the forearm toward or away from your body. It doesn't allow any twisting movements. These functions limits are imposed by the internal structures of the joint. So the way that ulna and radius articulate with the humerus determines that function of that joint. So another joint that was very similar to our elbow joint is our knee joint. It's also a hinge joint. When you move your foot or your calf toward your body is creating that hinge joint, like moving your forearm toward your humerus. In comparison to our shoulder joint, which is a ball and socket joint, it has a wide circular range of motion. We can move it in many different planes. So comparing those two shows that structure of that joint and how different they look in comparison to their function and how they're able to work with our body. Okay, so some basics from high school biology. All right, so we have characteristics of all living things. All living things have cells. Cells are our smallest structural and functional unit of all organisms. All living things are composed of one or more cells. So all living things have cells. If they don't have a cell, they're not a living thing. So all living things have cells. The organization. All living things have a structural arrangement which is a more complex than non-living things. This ranges from the chemical level to the more complex organism level. The responsiveness of living things or cells or tissues, the ability to detect and respond to stimuli, which are things that can cause a reaction in their immediate environment. For example, if we touch a hot plate or a hot stove, a hot oven, what is our reaction? Our reaction is to pull our hand away very, very quickly. So we all have responsiveness. Those cells are going to respond. Our neurological cells are going to say, nope, that's hot. We don't want to touch that. Quit. Then we have regulation. These are mechanisms to maintain a relatively stable internal environment as external conditions fluctuate. This is called homeostasis. So all living things have homeostasis. Without homeostasis, we wouldn't have that regulation. Our body would either be really, really hot or really, really cold. And when those extremes happen, cells start to die. So our body is naturally going to regulate it because it wants to maintain life. The most common example of homeostasis is our temperature regulation. Then we have growth and development. Growth is the increase in size as it occurs as part of development. Development is the natural progression in physical maturation. Examples of this are when a seed becomes a plant or when a baby becomes an adult. Our next characteristic of life is reproduction, which is the process by which organisms produce offspring. But then we have metabolism, which is the essential chemical process that takes place in living cells and organisms. This includes anabolic and catabolic processes, which is building up and breaking down all of those molecules in the body to create energy. Without energy, our body would not be able to go. We would not be able to sustain life. We wouldn't be able to sustain that homeostasis. We wouldn't be able to reproduce. So there's so many different things that metabolism is good for. Then we have on the other side the four processes of life. All four of these are common to all living things, and they all require energy. Excuse me. The first is respiration. Obviously, respiration is breathing. It's a fundamental process of life which involves energy production. Oxygen is obtained from the environment and it's required through a chemical process. It releases energy, and then we exhale carbon dioxide as its waste product. Then we have digestion, which is a mechanical and chemical process. It converts nutrients from the ingested food we take in, so the body can use it. It breaks it down into more simpler molecules, and then those molecules once again can be used for energy sources and sustaining life. Then we have circulation, which is the internal movement and distribution of oxygen, cellular waste, and the products of digestion. It's transported obviously by the cardiovascular system, which includes the heart, your arteries, and your veins. And lastly, we have excretion, which is the process in which undigested food and wastes of metabolism are eliminated. So the excretion can be either in the form of solid waste, which is our stool, or in liquid form, which is our urine. Now we have organization of life. They are all interdependent on each other. So it's a building block process. We're building one on top of the other on top of the other. Without the lowest chemical level, we cannot have an organism level. We must have that basic starting level with that chemical level. So throughout this semester, we are going to address different levels. And at the end goal, if you continue on to AP2, the purpose of AP2 is to kind of tie it all together and create that finalization of that organism level and fully understanding how the human body works. So obviously each level is more complex because they keep building on top of that level below them. Okay, so we're going to start by reviewing the chemistry and cells. This is in Unit 1. So that's our first cover because that is the basic level, the starting level. So we can apply this knowledge in each subsequent unit to better understand the tissue, organ, and system levels. So we're going to keep building on throughout the semester. You're going to see a lot of redundancy in regards to going back to basic concepts. So it's really important to fully understand this first unit because it is the building block for further units down the line and throughout your career in anatomy and physiology. Okay, so let's go ahead and dive into that chemical level. The chemical level is composed of atoms and molecules. Atoms are our smallest stable units of matter, and our molecules are made up of bonded atoms. So our atoms is the smallest, most possible chemical level you can get, and we're going to combine those atoms to make those more complex molecules. We're going to learn about different types of bonds, which are covalent, ionic, and hydrogen bonds, which occur throughout our body. The way a molecule behaves is determined by the shape and the anatomic components, and this allows us to make predictions about that molecule and how it's going to work in our body and what it's going to do. So, for example, enzymes are special molecules that catalyze chemical reactions. Their shape is very important to their function. If the shape is changed, they will no longer function. This is called denaturation. There are 4 different categories of the macromolecules that we're going to learn about as well. They're the carbohydrates, the proteins, lipids, and the nucleic acids. So we're going to get further into this as we go through the unit. We're going to talk more in depth about these different types of molecules, different types of bonds, and the 4 categories of those macromolecules. Then we have our cellular level. Cells are the smallest living unit. Human cells differentiate and specialize. We have roughly over 200 different types of cells. The humans start as one cell and become trillions through mitosis, and we're going to cover mitosis a little bit later on as well. They are very tiny and they're measured in micrometers. Cells can work together, but each cell can also respond to its local environment independently. The cell's function largely depends on organelles, which is made of molecules. Each organelle has a specific function. So, for example, the mitochondria produce ATP by breaking down glucose in the presence of oxygen to supply energy to the cell for the process such as contraction or transport. So, for example, skeletal muscles are very large. Think back to our relationship with anatomy and physiology, structure versus function. So those skeletal muscle cells are much larger. They have multiple nuclei in them. They have striations in them. They look very different than compared to cardiac muscle, which only has one nuclei, or smooth muscle, which only has one nuclei as well. So why would skeletal muscle cells have multiple nuclei? Think of the job that skeletal muscles do. So using that relationship between anatomy and physiology will help you determine, okay, this cell looks differently from this one, so maybe its job is going to be a little bit different, or maybe its job is going to do this. Okay, so we have cell theory. Do you remember this from high school? You probably touched base on it briefly in basic biology in high school. So we're going to go over it again as well. It's an important theory to remember. So definitely kind of put a star on this next slide right here, the basic principles of the cell theory, because you will need to know it for future information. So cells are the structural building blocks of all plants and animals. Cells are produced by the divisions of pre-existing cells, and cells are the smallest structural units that perform all vital functions. So cells are that building block of any living organism, plants and animals. They are produced by the divisions of pre-existing cells. We get cells from mitosis from other cells, dividing of those cells, creating more cells, creating that tissue, creating that from that tissue level, now then going to the organ level, and then that organ level going to the organism level. So cells are going to build upon themselves. They're going to divide from themselves. And cells are our smallest structural unit that perform all vital functions. So remember those little tiny organelles that live in each cell. They have their own little environment, their own little world. They can be independent of each other, but they also work really, really well together to create that big organism. So this is a really important slide. I want you to keep this in the back of your head. Look back at it. Make sure you understand this cell theory. Cell function is also related to structure. So how does the shape of the cells relate to its function? So I kind of alluded to this on the previous slides when we were talking about those skeletal muscle cells. And what predictions can you make about the function based on the structure? So in the first left-hand side, we have smooth muscle cells. You look at them, those are really long and slender. So smooth muscle, the job of smooth muscle, they are involuntary. They help with contractions of the walls of the stomach and the intestines, and they support the digestion and movement of ingested solids and liquids. So that's why these kind of look the way they are, because they're involuntary. They're constantly moving. They're constantly working. So they need to have that fluid motion. Other organs that might have some smooth muscles in them, those are the bladder, the uterus, our blood vessels, our trachea, our bronchi. So think of these things, think of the structure of these things, and think of what their function in our body is, and that can help you determine what that cell structure may look like as well. Then we have our red blood cells and our white blood cells in the next little section. Our red blood cells are more of a flattened type disc because their job is to transport that oxygen and carbon dioxide in the bloodstream. The white blood cells are a little more spherical because their main job is to fight off infection and to combat disease in our body. Then we have the next one is our bone cells. Those bone cells kind of look like, to me, they look like little tiny bugs or like centipedes or something with all those little fingers sticking out there. Bone cells are within small cavities inside the mass of the bone, and they help maintain bone and recycle the calcium and phosphate stored there. So if you actually took a cross-section of a bone and looked at it, bone is not solid. So looking at these bone cells, you can kind of determine that bone is not solid. Bone is very porous. Bone, if you took a piece of chalk or a piece like a concrete block and you broke it in half, they are very similar to how the structure of bone is. They're actually designed after bone because bone is very, very strong, but it's not solid. So creating these little tiny holes or air pockets per se creates this strong bone mass. And so if you look at the structures of those bone cells, that can kind of give you an idea of how they interconnect to each other and create all those little tiny porous details, making that bone really strong. Our next one is fat cells. Fat cells are spherical storage containers. When more energy, aka food, is taken and then used, the excess energy is stored as fat, causing these cells to become larger and more abundant. So fat cells, we actually have a set number of fat cells. They typically, once we're fully developed, we don't start replicating more fat cells. So if we gain weight, we're not getting more fat cells. That's not how that works. Those cells that we already have balloon up and get bigger. If we lose weight, those cells are going to shrink and get smaller. So that's how our weight, our cells are going to fluctuate in their appearance depending on how much weight we have. Then we have the reproductive cells. We have the oocyte in our sperm. So our oocyte obviously is our egg, and our sperm is from the male. So obviously our egg is going to be egg shape, and because it carries all that important DNA, RNA that it was given by our mother, and then the sperm needs that swimmer to swim up the reproductive tract of the female to implant into that oocyte. Then on top of our reproductive cells, we have our cells that line our digestive tract. So you see how those cells are long and skinny, and they are kind of compacted in there. We want those like that because, one, they're very delicate. They help absorb our nutrients that pass through our digestive tract from the nutrients we eat, from the food we eat. But also we want those cells watertight. We don't want our intestinal, we don't want our stomach, we don't want it leaking. We don't want a little break in those cells, because if any of that waste that we don't want gets into our abdominal cavity or our bloodstream, it can be very toxic to our body and upset that homeostasis that we're trying to maintain. So we want that really close, tight-knit structure of those digestive cells. And then we have our nerve cells, which is the very last one, that large tree-like looking nerve cells. Obviously, nerve cells help process information. Thought, memory, consciousness, muscle control are all actions that the neurons and the nerve cells perform. So it's really important. It's a huge web of communication. You can think of it like the invisible web that we have in our world, sending all this information throughout our entire body. Okay, so then we go on to the tissue level. Tissues are composed of similar cells that work together to perform a specific function. So we had the chemical level with our atoms and our molecules, which those atoms form together to create those molecules. And then we moved on to that cellular level, where we have those organelles and we have a little environment in our cell. And now those cells, those like cells, are coming together to create this tissue level. There are four primary types of tissues. You definitely will cover this in lab. We have an epithelial, we have connective, muscle, and our nervous tissues. And we're going to divide each of these primary tissues down and dive more in depth in each of them. So this little diagram here shows how we have our extracellular material and fluids and our cells that combine together to form those tissues, which are those four different types of tissues. And then those tissues can also form together to form those organs, and then those organs all work together to form that organ system. Okay, so our first type of tissue is our epithelial tissue. This is the most common type of tissue in our body. It is found in so many different places. Our most common, when we think of epithelial tissue, the most common type of tissue we think of is our skin. But also we know it's found in our digestive tract, it's found in our lungs, it's found in so many different parts of our body. So it does form a barrier with specific properties. It covers exposed body surfaces. So for our intestines and our skin is the most common one that we think of. It lines our digestive, our respiratory, our reproductive, and our urinary tracts. And it surrounds internal cavities such as our chest wall cavity. And it lines our inner surfaces of blood vessels in our heart. And it also produces glandular secretions. So epithelial tissue, like I said, is found in so many different areas. It has so many different functions. It is really a very, very important tissue type. Okay, so then we have our connective tissue. It is diverse in appearance, but but all forms can contain cells surrounded by an extracellular matrix. This matrix is composed of protein fibers, ground substance, which is a liquid, and the amount and consistency of the matrix varies by the particular connective tissue type. So, for example, we have blood, which is more of a watery type matrix, and then we also have bone, which is connective tissue, and this is more of a crystallized matrix with a little bit of ground substance. So that little cartoon picture over there is just an example of what connective tissue would kind of look like. with all its different, it has obviously our extracellular matrix. We have our different types of fibers and our ground substance in there. We have the collagen fibers. We have elastic fibers in there. So we have so many different things. So the main job of our connective tissue is to store energy. Next, we have muscle tissue. It has the ability to contract forcefully. Its major functions are skeletal movement, soft tissue support, maintenance of blood flow, movement of materials internally, and stabilization of the body. We have 3 different types of muscle tissues. We have skeleton, cardiac, and smooth muscle. For the purposes of our Anatomy 1, we're mainly going to focus on the skeletal muscle, but be familiar that there are 3 different types of muscles. Skeletal muscle, it directly or indirectly attaches to the skeleton, and when it contracts, it moves and stabilizes the position of the bones in our internal organs. So it's really important with structural support, protection of our precious organs. Without our skeletal muscles, we would not be able to stand upright. We would kind of be a blob and flop over. Our cardiac muscle obviously is found only in the heart. It contracts and moves the blood throughout our vessels and through our entire body. Then we have, lastly, our smooth muscle. This is found mainly in the walls of our blood vessels. It's found in glands. It's along our respiratory tract and our circulatory and digestive tracts, as well as our reproductive tracts. Lastly, we have nervous tissue. Nervous tissue is specialized to carry information or instructions within the body. So the nervous tissue is our information highway. It's our web, for lack of a better term. There's 2 different basic types of cells. We have neurons and neuroglia. The neurons are our main nerve cells. They transmit that information in form of electrical impulses. And the neuroglia are our supporting cells of our neurons. These isolate and support the neurons, and they form that supporting framework. So they are like our little helper bees to our neurons. So the nervous tissue is also divided up into different types of nervous systems. We actually have 3 different types of nervous systems. For our course, you will need to know 2 of those 3. But be familiar that there are 3 different types, and in Anatomy 2, you will learn about that 3rd type. So for our purpose of our course, we're going to be learning about the central nervous system and the peripheral nervous system, and just know that there's the enteric nervous system as well in there. So the central nervous system includes our brain and our spinal cord, and then the peripheral nervous system includes all the peripheral nerves that extend out into our hands, our feet, our arms, our legs. This connects to the central nervous system, and it transmits that information from our tissues and our organs. Okay, so we have our organ level. This is the functional unit composed of more than one tissue type. So we're combining those multiple tissues, that connective, the muscle tissue, the nervous tissue, the epithelial tissue. We're combining those all together, or 2 or 3 combining together to form those particular organs. The function is determined by the structure and the organization of the tissues. For example, flat organs may work well for protection, for example, our skin, but a hollow organ or structure could work well for storage, like our gallbladder, our stomach, our bladder. Most organs have all 4 tissue types, like I alluded before. The heart has lots of muscle tissue for contraction. It's lined with the epithelial tissue. It contains nerves to regulate contraction, and it has connective tissue to hold it in place. Then we have our organ systems. So for the following slides, these will need to be learned on your own. We're going to kind of breeze through them really quickly, but make sure you go more in depth with them. Look at these slides. There really is a lot of information on these next slides that you need to be familiar with and understand, okay? So the organ systems are multiple organs that interact to perform a specific range of functions, usually in a coordinating fashion. There are 11 different systems in the human body. They all work interdependent of each other. So for our course, we're going to be covering the integumentary system, the skeletal muscular system, the nervous system, and obviously reviewing our foundational chemistry and cell content for all the systems. So I'm going to pause our video here. Like I said, the next slides you're going to need to learn on your own. Just go through them, review each slide, learn about each organ system and what it does, its main function, the structures that are included in the organ system. Okay, so those organ systems are ingumentary system, skeletal system, muscular system, nervous system, endocrine system, cardiovascular system, lymphatic system, respiratory system, digestive system, urinary system, female reproductive system, male reproductive system. Okay, now we're going to move on to homeostasis. Homeostasis is important for the stabilization of our internal environment. All living organisms, it is very vital for them to maintain homeostasis for survival. To stay alive, many values must stay within a normal, narrow range. For example, in the human body, it's important to have our blood pH, our body temperature, all maintained at a certain tight range. If our cells go above or below that, we can start to develop a disease. We can develop any type of autoimmune disorders, genetic disorders, illnesses. Our body automatically tries to auto-regulate it, like our AC. It automatically, we set it to a temperature, and it's going to auto-regulate it to keep it at that temperature of 75 degrees in the house at all times. So your body knows that this is what it's supposed to do, and it has all these fun checks and balances to try to maintain it there. If it can't get it there, that's when we have problems, and that's when these illnesses and diseases and even possibly death can occur. There's this fancy little thing called homeostatic regulation. That's kind of like that AC that I was talking about. It's a physiological process by which homeostasis is maintained despite changing conditions. So that's that checks and balances that your body's doing constantly to say, okay, my temperature is at 99.5. What do I need to do to get it back down to 98.7? Or my blood pH is starting to creep up. It's becoming a little more basic. I don't want it to go there. What do I need to do to get it back down to where it's supposed to be at pH of 7? So there's all these little checks and balances that your body can do to create that homeostatic environment. So some simple examples of homeostatic regulation, obviously our temperature control, that's the most common, that's the easiest thing that we think of because our body likes to maintain that nice, warm, cozy 98.7 temperature, varying a few tenths of a degrees here and there. Obviously our blood sugar control, people don't really think about that one as much, but our blood sugar needs to be maintained roughly fasting between 80 and 120. And postprandial, which is after we eat, it should not really go above, for non-diabetics, it should not go above 150. For our diabetics, we got a little more wiggle room just because theirs can't regulate as easily. So we don't like them to go above 180. And then the body's insulin kicks in and it starts to bring it back down to that normal fasting blood sugar between 80 and 120. So if you ever have the opportunity to check your blood sugar, may it be before you eat, after you eat, you'll notice that your blood sugar really does stay within that 80 to 120 range if you're not a diabetic. And then, of course, we have blood pressure regulation. Our blood pressure is a very easily monitored regulation that we can check. Obviously, we just check our blood pressure. We check it with a blood pressure cuff. We can go to Walmart, CVS, Walgreens. We can go to the doctor. We have all these different places where we can check our blood pressure. A normal blood pressure for an adult is 120/70-ish, give or take. So ideally, we don't want that blood pressure above 140 because systolically, because when it goes above 140, we know that the heart is working too hard. It's squeezing too hard because that systolic tells me how hard is that heart squeezing and pushing that blood out of there. And then that bottom number, that diastolic number, is the relaxation of the heart. It tells us how forcefully is that blood coming back into the heart when it relaxes. So we don't want those numbers elevated because then that tells us that there's something going on in the body that's causing that blood pressure to be a little bit elevated. And then vice versa. If the blood pressure is too low, it tells me that the heart is not squeezing hard enough and that blood, when it comes back into the heart, is coming in, kind of trickling in, and there's a kink in our hose somewhere. So homeostasis is an amazing thing, but it is very tightly regulated. All right, so we have our components of our homeostatic regulation mechanism. There's that pretty little thermometer that I was talking about. So we have our receptors, which are our sensors. The sensor is something that is it senses change in the condition, which is our stimulus. Then we have our control center, which is our integration center. This center processes information from our receptors, which is our sensor, and it sends out commands for what to do in response to that stimuli. And then we have our effector. The effector responds to the commands to address the stimulus. So like I said, we're going back to that AC in our house. So maintaining the temperature in your house, the receptor is your thermometer. So your thermometer checks the temperature in your house. Okay, where is our temperature at? Is it at that set temperature that we have, or is it above or below? That control center is also the thermostat, and it's kind of the AC unit. And then that effector is the air conditioner. So if our receptor, our thermometer, detects that we're getting a little hotter in there, that control center, that main thermostat is going to kick on. And the effector is going to be notified. It's going to be, I'm not an AC person, so it's going to be kicked on to say, hey, we need to blow cold air because it's getting too hot in here. We need to make it back down to that 75 degrees. So that's how that works. And then once that thermometer, that receptor identifies that, okay, now we're at 75 degrees, it tells that control center, hey, okay, we're done. And the effector shuts itself off. So that's the same concept how our body works. So with our temperature regulation, if our body temperature goes too high, we start to shiver to kind of cool ourselves off. And then, reverse that. I apologize. So if our body temperature goes too high, we're going to start to sweat to cool our body temperature down. But if our body temperature goes too cold, we start to shiver to help I apologize, tongue twister. Our body starts shivering, so those muscles start to create heat and energy to try to warm our body back up. Okay, so obviously this homeostatic control is not precise. So just like your AC, it kicks on because it regulates and says, hey, it's getting too hot in here. It's not 75 degrees, it's 76. So then the AC kicks on. And I don't know about you, but when I'm at my house, and if you're sitting under the AC vent, even though the thermometer says it's 76 degrees, when that AC kicks on, it's blowing really cold air. So it feels like it's dropping really, really fast. And it could actually be cooler in the room than the temperature is saying because it's trying to get that temperature down really, really quick. So you've got those fluctuations up and down, up and down, up and down of the air, making it feel colder or making it feel hotter in your room. Same thing with your body. It's going to fluctuate up and down because it's never going to stay exactly where it's supposed to be because it's trying to keep that average temperature where it should be. All right, so now we have negative feedback. This is the most common mechanism to maintain homeostasis. The effector opposes, remember the effector is that sensor, or negates the original stimulus. For example, if the stimulus is feeling too cold, then the negative feedback would result in getting warmer, which is the opposite of the stimulus. This minimizes our change, and the set points can vary with different environments and activity levels. So negative feedback is the primary mechanism of homeostatic regulation. It provides that long-term control over the body's internal conditions and systems. Normally it ignores minor variations and it maintains a normal range than rather a fixed value. So it may have the range for your temperature may be anywhere from like 97.8 degrees up to like 99 degrees. That may be your normal range. Everyone can be a little bit different from person to person. For example, the dynamic process, the body temperature can vary moment to moment or day to day. So when you sleep, your temperature set point is a little bit lower, because you're not as active, you're not moving around as much, so you're not generating more heat with those muscles, versus being outside on a hot day. Of course, your natural resting temperature is going to be a little bit higher because your body's working a little bit harder. It's hot outside, your body's trying to work itself to cool it down a little bit more, so your body inside is working a little bit more, so it's going to generate more of that heat. Other examples of negative feedback include obviously blood sugar regulation. So the more carbs and sugars you eat, the more your body produces the insulin to lower your blood sugar. And then obviously the less carbs and sugars you eat, your body doesn't need to produce as much insulin, so it's going to give you a little bit less insulin to lower your blood sugar. Your blood pressure control, so they're called baroreceptors inside. They help detect the changes in your blood pressure, and then it sends the signals to the brain to adjust the heart rate and the blood vessels diameter. So it has those blood vessels constrict and dilate depending on what your blood pressure is doing, if it's high, if it's low, to help regulate that blood pressure to try to keep it in that 120/70 range. So this is just a little diagram picture of what we just talked about. It's a great diagram. It talks about where homeostasis is, how the stimulus affects the receptors, how that control center sends the information saying, hey, we need to autocorrect, our temperature is getting too low or too high. What are those effectors going to do? What are the effectors? Like the effectors are going to be those sweat glands because we're going to start sweating. The smooth muscles in the walls of the vessels may be contracting to help stimulate heat. And then restored homeostasis occurs because now we've either released those sweat glands to cool our body down, or we have contracted those muscles to create a shiver to generate more energy. All right, so there is another type of feedback, which is positive feedback. This is less common. It doesn't happen very often, but it's still important to understand what is positive feedback and how does it work. So the effector exaggerates or enhances the original stimulus. And you think, why would I want to do that? Why would I want to create more and more and more of what that sensor is detecting? It is important because this produces extreme responses. At times, it's really important to have that extreme response. So, for example, we have labor contractions. If we used negative feedback for labor contractions, we would never be able to birth that baby. That baby would be stuck in there because our body would say, "Oh, nope, you're contracting too forcefully. We need to calm down. We need to relax," and it would relax that muscle because that muscle's working too hard. So for labor contractions, we want those muscle contractions to build up on top of each other, stronger and more forcefully until we're able to birth that baby through the birthing canal. So the first contraction leads to stronger and more frequent contractions, taking us away from the homeostasis, but ultimately the baby is then born. So then we return to homeostasis, correct? Because we no longer need those strong contractions of our uterus to push out that baby, that fetus. Another really important example of positive feedback is clotting. Without positive feedback for clotting, we wouldn't be able to clot, we wouldn't be able to form scabs, and we wouldn't be able to heal. So a cut or injury to a blood vessel leads to accumulation of platelets, which bring even more platelets, and then that leads to the clot that then prevents the blood loss. So then we eventually get back to homeostasis because the body recognizes, now I have my clot. I don't need to continue to send all these helpers down there to create that clot, so I can turn that off and we can create that clot and create that scab and start to heal. Another example of positive feedback is lactation. When the baby suckles, it causes the production of prolactin, which then causes the milk production. The more the baby sucks, the more milk production we get. So a lot of women, if any of you have ever had children, you understand this concept. It's your baby latches to you, or even you see your baby, your body naturally sees, gets that urge and gets that release of that prolactin, and then you get that milk letdown. So that is a positive feedback example as well. So this is a diagram of what would happen with an injury and how our clot would form. So definitely review this slide. It's really a great slide. It talks about that positive feedback loop and how, like I said, how we create that blood clot. Okay, so this is something that you're going to be really focusing on in lab, but I love this slide. It's a great slide. The next couple slides are wonderful. And anatomical terms are really important. So please definitely study this. Know your anatomical terms because you will use them throughout the rest of your medical career, may whatever it be. These are things that are used to describe things. So it doesn't matter if you're reading literature, if you're reading chart notes, if you're describing something to a fellow provider, fellow nurse, anyone, they know where you're talking about it, and there's no question about this is the location. So for anatomical position, when you stand, there's two different standard anatomical positions. You have standing up and you have lying down. So when you're standing up, your hands are at your sides, your palms are facing forward, your feet are together, and your toes are pointing forward, and your eyes are facing forward. So I encourage you to kind of pause the video right now, stand up, and stay in an anatomical position. Usually for me, I'm a doer. I have to draw pictures, or I have to physically touch it or do it to fully understand it. So if this is a way that you learn, I encourage you to do this because it will help you fully ingrain in your memory, okay, what is anatomical position? You will be asked, what is anatomical position? I promise you, either in my class or in lab, you will be asked what is anatomical position and describe anatomical position. All right, so then the other one is lying down, which is similar to above, except obviously you're supine. You are face up and prone. So when you're supine, you are face up, and when you're prone, you're face down. So both of those are anatomical position. It's the same thing. Your palms are face forward, and your toes are pointing forward as well. So if you're supine, which is face up, your palms are up and your toes are pointing toward the ceiling. If you're prone, you're face down, your palms are pointing toward the bed. Let's say you're laying on a bed. Your palms are pointing toward the bed and your toes are pointing toward the bed as well. Okay. So here's also some directional terms that you should be really familiar with. You have superior and you have inferior. So superior means just toward the top, and inferior means toward the bottom. It's kind of a generalized term. You can use it kind of interchangeably with other things when you're describing something. So you can say, for example, the head is superior to the shoulder, or the knee is inferior to the hip. So that's kind of how those are used in descriptive terms. Then you have your lateral and your medial. So lateral is away from the midline of the body. So if you cut yourself down, or again at the next slide as well, we'll have some of those descriptive terms. If you took a line and you drew it between your eyes, through your nose, all the way down the center of your chest, to your breastbone, through your belly button, all the way down that way, that is your midline. So lateral is away from your midline and medial is toward your midline. So this can be used anywhere. You can say the lateral aspect of the foot, the medial aspect of the foot. So the lateral aspect of the foot is going to be the pinky toe side, and the medial side is going to be your great toe side, your big toe side. You can say the lateral aspect of your face, the medial aspect of your face. So these can be used, the lateral aspect of your abdomen, the medial aspect of your abdomen. These can be used anywhere on your body to describe, is it away or is it toward? Then we have proximal and distal. So proximal is away and reverse. Proximal is toward and distal is away. So it's almost similar to that superior and inferior concept. So proximal is going to be toward the top. So you could say your knee is proximal to your foot, or your wrist is distal to your elbow. So those are going to be examples of using the terms proximal and distal. Usually we use proximal and distal more for your appendages, and then superior and inferior when we're talking about general overall body stature. So, and then we have the lateral view picture. We have cranial or cephalic, which means head. And we have caudal, which means tail bone or tail. So the cranial is another way of saying superior, and the caudal is another way of saying inferior. And then we have our posterior, which is our back, and we have our anterior, which is our front. Posterior also has the term dorsal, and anterior also has the word ventral. So here's those sectional terms I was telling you about before. We have our frontal or cranial planes, where we cut our body down. If you look at the picture, it's that purple slice there. You're basically slicing your anterior from your posterior. Then we have our sagittal plane. You're taking it and you're running it down that midline of your body. That's another way in saying the midline. You have your mid-sagittal, which just means that you only go to the waist. And then we have our transverse plane, which is that green one there. And that's just cutting us right in the middle of the body, kind of right around the hip area, belly button area. So we're cutting it in half. So the transverse sections can really be anywhere. When we get MRIs and CT scans, we usually cut in transverse sections, and that's how we view the body or view the appendage that we're looking at. We cut them in those different types of sections. So if you're going into radiology, these sectional views are really important because, especially if you're doing CTs and MRIs, you need to understand how do those work? How are they cut and how are they read? And different cuts are going to give you different views. So obviously, if I am doing a CT of our chest, and I start at the lower portion of my chest and I'm working my way up, when I get closer to our clavicle bones or our heart, that view is going to look really different if compared to my lower area where my diaphragm is and the lower tips of my lungs are. So you definitely understand, depending on where you cut in the body, you're definitely going to get different images and views. All right, so here's just a little question. A surgical incision was made along the sternum to open the thoracic cavity for heart surgery. Along what anatomical plane was this incision created? So this is something that, helps you think about those different planes that we just talked about. These are type of questions that you may see on the exam. This is just like that, applying what you know, applying that structure to function kind of thing. So the answer is our median or mid-sagittal plane. So that's that last one down there on the bottom. All right, so like I said before, location of a section can alter appearance. So we need to think in 3D. We need to kind of place them all together to get that full picture of what are we looking at exactly. So this is super relevant to medical examinations such as CTs, X-rays, and MRI scans. But also in lab, you're going to study these structures with the microscope. So the different cuts of the whatever slide you get, you're going to get different views. So what you may see may not be the same thing that somebody next to you is going to be seeing. Or you may grab 3 or 4 different slides of the same thing and you're going to view different views each time. So just be familiar that, like the example here is the artery. Depending on where I cut that artery, I'm going to get completely different images of that artery. All right, so this is something too that you're probably going to see in your homework or your quiz assignment. These are these body cavities, okay? So this is something I want you to learn on your own. Like I said, it is going to be in your homework and your quizzes, so you're going to get some redundancy there. You're also probably going to learn this in lab as well. So just go through these, be familiar with the different cavities, which are your pleural cavity, your pelvic cavity, your pericardial cavity, your diaphragm, and your abdominal cavity. So just be familiar of where they're located. All right, and that is the end of our first chapter. I'll be back with you and we'll go into chapter 2.